Ann Summers is a professor of microbiology at the University of Georgia. She studies bacterial metabolism of mercury, the biochemical basis for cellular damage by inorganic and organic mercury compounds, and the mechanism of spread of antibiotic multi-resistance. She is a fellow of the American Academy of Microbiology, the American Association for the Advancement of Science, and the IAOMT. Her work is currently or has been funded by the NIH, the NSF, the DOE, the EPA, and several private sources, including the J.S. Guggenheim Foundation and the IAOMT. Please give a warm Academy welcome to Ann Summers.
Thank you very much, and let me first do this de rigueur recital that I do not have any financial interest of a product in my talk or with any companies offering grant monies for this continuing dental and medical education program. With that, thank you very, very much to the organizers of the meeting. It's good to see that you have such a great crowd here this afternoon, and I'm very encouraged. I went to my first IAOMT meeting, I think sometime in the early 90s. It might have been 91 when they were in Atlanta, and I've been to several since then. And from the very first, I was impressed with the energy and the dedication and the compassion of the organizers and the people who were attending the meeting. And at the time, I think just like many of us who were aware of the dangers of amalgam-mercury, it never occurred to me that the organization would last for 25 years. It wouldn't need to. It would all be over and done in a decade or less. And of course, that hasn't happened. And fortunately for the members of the association and the society, and also I think for myself and others who work in this area, our optimism is what's carried us beyond the 10 years, which we thought would probably be all that was necessary for an organization like this to exist. So you're still here, you're still active, you're still doing good things, and obviously very recently, all of this groundwork that you've heard about and know about from your emails and so forth in your presentations earlier today, all of that is really finally going through a threshold, I think, in terms of getting some action at levels beyond where we work, but they're finally beginning to listen to the kinds of things that practitioners and research scientists are saying about what a bad idea this is. So we're going to switch gears from the more reflective presentations and clinically oriented presentations that we've had earlier today, and get into some basic science, some science that underlies the phenomena that impinge upon your patients when they have amalgams. And I'm a bacteriologist. I'm trained primarily in chemistry and biochemistry, but I use as my experimental organism primarily E. coli, a microbe. And so that's the topic of this, and it actually goes way beyond E. coli in principle, as so much of human biology does now. It used to be that, less so in dentistry, but I think in many medical meetings, you had your internal medicine folks, you had your radiology folks, you had your cancer biology guys, oncology guys, and everybody would have their separate meetings and rarely get together. And the infectious disease people were really always on a limb. I mean, most of the functional disorders that people are aware of, they don't think of as infectious diseases. And so there had been this separation between understanding the biology of the organisms that live with us and in us and on us all the time, and this, that, or the other disease. It was as if they were completely separate worlds. And you may know that in medicine generally and in medical biology generally, there has been a great epiphany, an awareness over just about the last 5 years, where there's a recognition that microbes is us, okay? The microbes that live in us and on us outnumber us by 10 to 1 in terms of the cells of our own bodies. We are a community, every single one of us, and a very varied community, not identical, more unidentical in a way than our own genes would make us seem. And these genes of ours and the genes of our bacteria work together. It's going to be a real challenge to unravel all of this in service of human health, but it's a very important task to do. Sometimes human history leaves us with something that seemed like a good idea at the time, kind of thing. to repair something that, in fact, another aspect of human history had also found seemed like a good idea at the time, and that was sugar. Sugar wasn't a part of the diet of humans very much up until the 19th century, and then it became more abundant on a large scale. It's not that people didn't overeat sweet things, which they did, but natural sugars don't have the propensity to cause caries in the way that pure sugar does. And for a whole lot of economic and social reasons, sugar came on really strong, and also did the need to take care of folks who were losing their teeth because of sugar. And as all of you know, one of the mediums media that was invoked to do this was dental amalgam silver. And I don't need to rehearse the observations that you've heard from others today already, but I do have a few things I want to say about this. And I do want to start off, first of all, acknowledging the folks whose work I'm going to talk about today. There's my group at the University of Georgia, which includes both my students and my colleagues in various other departments that I'll refer to. And then University of California, San Francisco, Sue Miller's laboratory and mine has collaborated for almost over a decade. And she's a biochemist, enzymologist at UCSF. In some of our very recent work, we're collaborating with folks at the Pacific Northwest National Laboratory, the Department of Energy. And also, in very recent work, we've collaborated with Chris Shade at Quicksilver. Funding for these various activities has been supplied by the U.S. Department of Energy, primarily in my own lab now. And we've also gotten funding over the years from the IAOMT. I apologize for this counterfeit logo. I couldn't clone your logo off the website, guys, so it's the best I could do. But we've also received funding from the University of Georgia Research Foundation, or the college, Franklin College of Arts and Sciences. So, whoa, I don't want to shut down my computer. Stop that. Thank you. Okay, the outline of this talk is as follows. First, I'm going to talk about some old work where we actually began working with IAOMT members, Marie Bimmy and Fritz Lorscheider years ago, that showed that the installation of amalgam affects the bacteria of the mouth and the colon in a way that is not benign and led to some concern in a completely unrelated area of infectious diseases. Then I'm going to talk about a surprising, but it's not really surprising to a microbiologist, but perhaps surprising in the general public, is that intestinal microbes can methylate mercury inside when provided with mercury from dental amalgams. And lastly, I will talk about no, okay. See, I wanted to use my own Mac, but it's betraying me. The exposure of bacteria to various inorganic and organic forms of mercury leads to the formation of stable mercurial adducts on very important proteins. Now, to any biochemist, that would be like, oh, ho-hum, we knew that. Okay, why is this important? What's important, I mean, it must be important because absolutely nobody's paying any attention to it, okay? And that's not quite true, but it is in a sense true, because people who work on organ systems of various kinds, and I'll give you an example in a moment, don't tend to think about mercury because they know it's toxic. And they also don't know how much exposure there is to it in the human population. Not just from, they know about methylmercury from fishes, but that's about it. And so the story I'll tell has to do with the folks who discovered the role of the cysteine, sulfur amino acid, in the water transporter. There's every cell in your kidney, every cell in your body actually has a very important water transporter protein. And it's been almost a dozen years ago that it was discovered that in humans, that protein has a cysteine residue in it. That's sulfur. And I'll come back and tell you a little bit more about that later on. Not that protein, but sulfur and cysteine. And there was a paper published in the Proceedings of the National Academy of Science, which really excited me. It was from this group, an prominent Eastern university, that showed that if you treated cells that contained the version of that didn't contain the cysteine, cadmium didn't bother them at all. Cadmium is very closely related to mercury. But in the human version of it, the cysteine alone was responsible for giving very erratic behavior of that protein in the cell culture system that was examined. And they also referred in the paper to having examined mercury, but they didn't say anything further about what mercury did. So I called the guy and I asked him about what happened when you gave the cells mercury. And he said, oh, it wasn't really very interesting. And I said, what do you mean? He said, well, they didn't do anything. I said, that's interesting. Okay, so there is a gap between interests. We all get pretty narrowly focused in academia, and it's inevitable. So the last project today that I'll tell you about is what we're doing to actually look at proteins in the cell that are at risk from mercury exposure. It's not every protein, but there's some very serious players that are important. Okay, this is a fairly large, this is just a slide that I cribbed from the U.S. Geological Survey from its website. It's a big book that they publish every few years on materials flow and mercury in the economies of the United States, and this goes back to 1970 and up through about 1998 is this particular graph. And this is metric tons of mercury used in various applications. And this is batteries, and you all remember that in the late 80s and early 90s, we quit having mercury in batteries. And that made a big drop, okay? And also in paint, and that was a good thing. And these were organomaterials that were used as preservatives in paint, and so that made a big drop. The yellow part here goes way down, and the stripy part is batteries. Excuse me. And then there are other little things that kind of went, instruments kind of quit using them quite as much. It's kind of hard to see here, but the purple is dentistry, and it flickers along, and then when all the others drop down, it drops down too. But it still remains a fairly thick component up here even through the 90s. So while mercury in dentistry is not a major part of world use of mercury, it remains a fairly substantial chunk of what mercury we now do use. Now, the good news is that we quit using quite so much mercury. The bad news is that most of this is still out there, okay? Some of it's, obviously some of it's recovered, but there's the residue, what escaped during the time we were using these very high amounts is still out there. So we do have generally a fairly serious environmental contamination problem, apart from the internal contamination problem that we experience, we have fillings. So this is a mercury cycle. And I'm going to draw it on two levels here. This is the whole ecosystem. Mercury is released from industrial processes, and you get local contamination. You also get atmospheric contamination. And that falls back down and contaminates lakes and oceans and so forth. And within these aqueous bodies, microbial processes carry out various transformations of mercury. There is no form of mercury that is beyond reactivity in any biological system, okay? Mercury in any of its forms is transformable to its other forms by some biological entity. Bacteria, hands down, do it all. Oxidation, reduction, methylation, demethylation, okay? I'll show you on the next slide. But we do some too. Higher organisms can also carry out many of these processes. But the ones that are believed to be major players in what's called the global cycle are bacterial. And here's the fish. They can get methylmercury produced by bacteria, and then up here at the top of the food chain is where you build up that mercury. Now, good evidence says that the major methylators of mercury are, in fact, in fish in their intestines. They're not in the water column necessarily. They're probably part of the commensal flora of the fish. So the fish get the mercury, and then their intestinal bacteria can convert it in many cases that have been in one case it's been examined, in other cases that have been tested indirectly for methylation. So the bottom line for this business, or the top line, is that the global atmospheric burden of mercury is 1,000 metric tons, which is quite a lot of mercury. It has a half-time in circulation of about 1 to 2 years, which means it gets up there and then it comes back down again. So it's not like in a reservoir up there in the atmosphere staying put. It's coming back down to Earth. So we continue to use mercury, it will continue to go up. This is taking a while to switch, and I'm not quite sure why. Oh, there we go. Okay. So the US fossil fuel industry puts out 46 metric tons. So coming from industry, the burning of coal, puts out about 46 metric tons. And also the industrial chlor-alkali processes, isotope processes, batteries, and dental offices are responsible for local contamination. Chlor-alkali and various isotope production and batteries are all regulated. Fossil fuels right now, there's millions of dollars being spent, and they are regulated. The purging that has to be done, the scrubbing of the output is a heavy-duty expense on the US energy industry. Dental office use is not regulated much. It is more in Europe than it is in the States. Okay, so okay, there we go. All right, so this is a small size thing. The people aren't mentioned in here, okay? We get as far as fish over here, but no mention of the people. So now we're going to mention the people. The people that work in the offices where mercury is used can be exposed. The people that do the various industrial tasks associated with the use of mercury can be exposed. And obviously, from the fish, human beings that go to eat in restaurants or buy fish can be exposed. What's not mentioned in this particular rendering of it is the fact that red spots, meaning mercury, are present in all of these folks, or many of them, throughout the world. So what about that mercury? When I made this slide, it was about 30 metric tons of mercury that's used in dentistry in the US every year. Compare that to the 46 metric tons that is thrown into the atmosphere by the entire US fossil fuel industry. Except that's going into the atmosphere. That's 30 metric tons that's being installed in people's mouths here, okay? The average number of amalgams was 10 at the time I did this, which would mean, since the filling has about a half a gram of mercury, that would be 5 grams of mercury per person in their mouth on the average person. And they leak about a microgram per gram per day. Now, if you, at the time, there were around 300 million people in the United States, and the estimate was that about 80% of them had some fillings, and I was using this average number for that. And that would mean that there were 1,200 million grams of mercury in US mouths around the world. Actually, a couple of years ago, Bank of America did a higher number. I came up at this point with 600 metric tons of mercury walking around the United States inside people's mouths. Okay? Now, I told you all this other stuff is regulated over here, all right? Atmospheric release, all the other industrial things are regulated. So if the government regulates something, you can be sure that it's dangerous. And if they don't regulate something, then you've got to assume it's safe. And the government, de facto, or in effect, has declared that the only safe place to keep gram amounts of mercury for decades on end is in your mouth. Okay? This is part of the problem, okay? As I'm sure all of you who are aware of the political side know. It seems unlikely, I know. If you're going to regulate stuff over here, why not regulate it over there? Because after all the regulation that takes place over here, presumably, is to prevent any exposure from these people to mercury, or any animals or anything else. Okay, so this is one of the paradoxes that remain in this field. Okay. I want to just quickly, this is, in theory, a less busy slide than the big gray one was. This focuses down on that biotic cycle that can be carried out, and soluble mercury is the major culprit. This is the thing that's most dangerous. It can react with the proteins in our cells, as I'll show later, and it can make them inactive. Now, mercury zero, the vapor, is claimed to be, or the elemental mercury, the liquid mercury, is claimed to be non-toxic because it's inert. It has all the electrons. It's zero valence, which means I got all the electrons I need. I don't need to snatch any electrons from your proteins, okay? And I'm going to volatilize anyway. So this mercury is argued to be non-toxic. In fact, it's not inert. In chemistry, everything is only as inert as the next strong catalyst it runs into, okay? And I think Jess was saying earlier, talking about equilibria and so forth. Well, there's plenty of strong catalysts in our bodies that can handle Hg0 in a twinkling and convert it to Hg2+. Those are enzymes called peroxidases, primarily the heme peroxidases. Okay? An example of that is lactoperoxidase, which is a dominant enzyme in saliva. Okay? It's part of our protection system, and it is a heme peroxidase, and it can make a ferryle radical that just peels an electron off of, off of mercury in a twinkling. Okay? So we can make mercury more toxic. This is part of the biotic cycle. Hg0 can become toxic by reactions that carried out in our own bodies. Okay. Methylation. Okay. The substrate for methylation is mercury 2+, inorganic ionic mercury. All right. The cofactor, cobalamin, that's vitamin B12. We need it for our metabolism. We have bacteria in our gut that make it. We can use it absorbed from nutrients in our food and so forth that we take. Cobalamin is methylated in our cells during a process that we use to make amino acid methionine. That's part of our biochemistry. We can do that. Methylated cobalamin will methylate mercury. Okay? That was done in a test tube over 30 years ago by a chemist at University of Illinois, John Wood. Okay? You just get methylcobalamin and put mercuric mercury in there, and you got methylmercury. In fact, that's commonly the way methylmercury is synthesized in research labs. Okay, so this is a process we can carry out depending upon how much methylated cobalamin we happen to have, or how much methylated cobalamin our bacteria happen to be using. Now, sulfides, this is sulfur S minus, can precipitate mercuric mercury. And it is an insoluble form of mercury called cinnabar, bright red stuff. But sufficiently high acidity can resolubilize it. So if you have a place that has very, very high acidity, which some mine settings do and some industrial settings do, you'll get Hg2+ back again. So all these things can be carried out. The ones that I've drawn with the black arrows can be carried out by our biology. Not so much this one, actually, though. There are possibilities for this, again, in the intestine, because we have bacteria in our intestines that make sulfide. But all of these things can be carried out in the context of our biology, microbiology, fish biology, bear biology, not so much plant biology. Plants are a little smarter. They do some different things. Now, the things that I have spent the large part of my career studying are these two enzymes and their helpers. An enzyme called MER-A, which is capable of taking mercury 2+ back to mercury 0 and volatilizing it, therefore detoxifying the milieu for the bacteria. And then an enzyme called MER-B, which will strip the methyl group off of methylmercury, hand it over to mercuric reductase, and mercury 0 blows away. So if you're a single cell organism, this is great, because you're out there floating around in the in solution or in a biofilm or something like that. And if you have this enzyme, you simply reduce the mercuric mercury when it comes along, and it blows away. Higher organisms, metazoans, anything multicellular, doesn't have this detoxification system, which I'll mention a little bit more about in a minute. Strictly a bacterial thing. Okay? So bugs came up with this as a way of defending themselves against mercury, and it has led us to understand many things about how they experience mercury. And some of these enzymes, in fact, have been recruited by genetic engineering to help detoxify sites where there's mercury contamination. But they're bacterial enzymes that can do it, not ours. Okay, so this is the system that I work on. It's found in the bacteria, as I said, and this is a typical bacterium, and that's its chromosome, a circle of DNA, and it's carried on a plasmid, this resistance to mercury as it's called. Plasmid is a little extra chromosomal circle of DNA that carries genes that the bacteria might need sometime but doesn't need all the time. It's the same place, as I'll show you, where antibiotic resistance genes are carried. So the plasmid that we study has a locus on it called a transposon, which is a mobile genetic element, transposable element. And when we began studying it, we were primarily interested only in the mercury resistance down here. okay? And it's called an operon, a suite of genes that does a job for the cell. There's MER-A, and sometimes MER-B is present in this same array. And they carry out the reactions I just described. Mercury 2 plus gets reduced to Mercury 0, and MER-B removes the methyl group from mercury yielding mercury. So these are the players. And what we only came to pay attention to when we began working with Fritz Lorscheider and Murray Vimy in the late 80s was something we knew, but we weren't paying attention to, was that the other genes in this transposable, this mobile genetic element, are for resistances to antibiotics, several different antibiotics. So here's a resistance to sulfadiazine and resistance to quaternary ammonium disinfectants, which are fairly common disinfectants. This is a resistance to streptomycin in this case, but there can be many different kinds of resistance genes plugged into this particular array as it occurs in millions of different settings that have been, well, thousands have been examined, different bacteria worldwide, because this is a very popular, if you will, mobile genetic element, and it resides on plasmids that are very promiscuous, because plasmids not only hang out in one cell, they can actually move to other bacterial cells, and they take the genes along with them. So this right here is a demonstration of, or an example, an instance of, called genetic linkage. These genes are physically on the same chromosomal or same genetic element, the plasmid element. Okay? So exposure, and I'm not going to go through all this because this has to do with the enzymology, which I can talk to you about later. And I will I'll only say here that this is this set of genes that I just whizzed by here is actually found in a great many different bacteria, in gram-positive bacteria and also gram-negative bacteria, and in particular, things like Staphylococcus aureus has mercury resistance. In fact, very, very frequently has mercury resistance. Pseudomonas aeruginosa and Shigella flexneri, which is a close relative, actually, of E. coli. So these very common and frequent pathogens have a locus varying only slightly in its components to the locus that I just showed you. And in every case that's been examined in the gram-negative bacteria, and a great many cases have been examined in gram-positive bacteria like Staphylococcus and also Streptococcus, where the mercury locus is on some kind of transmissible genetic element that also carries antibiotic resistance. So, well, how did that happen? Okay, when we first saw this, we thought, oh, well, you know, antibiotic resistance is selected for all the time in people, and humans get it all the time, and the mercury resistance must just have gotten on there maybe when the bacteria were going through sewage treatment or something, because there was mercury in the sewage or something. But the studies that I'll tell you about in a minute actually tested that question, and we found out quite otherwise. But the point is that the antibiotic resistance goes along wherever the mercury resistance is. Okay, so way back in my first interaction with IAOMT began because we were interested in, it didn't begin at this time, but it began as a result of these studies. We were interested in how prevalent mercury resistant was in the commensal microbiota. We were working with Stuart Leidy, who's well known for his work on antibiotic resistance, and he had a huge population of human beings, some hospitalized, some not, from whom he was getting oral samples and fecal samples, and he was screening for antibiotic resistance genes. And he sent us some of those samples. In fact, we shared all the samples, and we screened them for resistance to various metals. And indeed, far and away, the most common resistance to a metal in all these human samples, it was present in as many as 60% of the people, and it was prevalent in about 30%. And this gives you an idea of this. It's actually not incidence. We did that in a particular time. It's actually prevalence. So it's the percent of all subjects, the percent of everybody that we looked at, and we divided them into decadal divisions as to whether they had 100% of their bacterial DNA, bacterial strains that we looked at were resistant to mercury, or none, or that's actually supposed to be 1.0, or a tenth of them, or a hundredth of them, or a thousandth of them. So this is a profile of how many of their bacteria per person were resistant to mercury. And we also measured the antibiotic resistances that they had in their bacteria. This is a Petri plate exercise. Okay, so what you can see is that these two curves, or these two profiles, go in different directions. So let's just look at the mercury resistance. The mercury resistance increases. The mercury resistance increases across here. And so we looked at, what's plotted here are the people who had no antibiotic resistance in their bacteria and the people who had two or more. In other words, they were multi-antibiotic resistant in their bacterial flora. And they go in different directions. So the likelihood of having no antibiotic resistance at all is greatest if you have vanishingly small mercury resistance. Okay? And as mercury resistance increases, your likelihood of being antibiotic resistance free in your bacteria declines. Okay? Concomitantly, having two or more, in other words, being antibiotic multi-resistant community in your GI tract or your oral flora, increases as a function of whether or not you also have mercury resistance. Okay? So that made us think that maybe there was something else going on here, that it wasn't just a matter of exposure to antibiotics, that maybe mercury resistance was a player. So we studied these strains in many other ways over the years, and then one afternoon I saw an article from the FASIB journal, and it was the article that Fritz Lorscheider and Murray Vimy published on a sheep model system, where they had put amalgam fillings into a sheep. And I think they'd actually done a couple of sheep at that point. And there were these marvelous and this was radioactive mercury, so they could do autoradiography. They could actually see where in the organs of the sheep they were. The mercury had gone. And I just thought this was great. This would tell us something about, this kind of a model could tell us something about antibiotic resistance, because there was clearly a lot of mercury in the GI tract and in the mouth, where our bugs had come from. So I called Fritz, and I asked him if he'd ever done anything with bacteria, and he said no. And we talked, and he said, we're just about to begin to do some monkey experiments, and would you like to participate? And we agreed to do that. And we went through a total of 8 monkeys altogether over the years, and these are the middle couple of them. And this is typical of the experiment. The monkeys would be in the vivarium up in Calgary for a period of time with nothing being done to them, just fed antibiotic-free food. They were always fed antibiotic-free food and fresh vegetables and so forth. And so this part here is the pre-adaptation period. This was kind of short for these guys. And then on day 0, the fillings go in, and we keep the monkeys for X number of weeks in these 2 experiments. And in this experiment, the monkeys were kept in-house with fillings for 2 months, and then the fillings were replaced with a glass ionomer filling. And what they sent to us from Calgary were oral swabs and fecal swabs. And we did our bacterial plating thing with them. And we also got mercury measurements on them from Calgary. And this is what this is. This is where the mercury is in the feces. So this is from those 4 monkeys. And there's our post-mortem numbers on the mercury they had in their kidneys post-mortem. But you can see, this is a log scale. This is 0.01 molar, okay? And it goes all the way up, 0.01 micromolar. So this is a very, very low amount of background mercury before the fillings go in the feces. And it goes all the way up to 1,000 micromolar, which is 1 millimolar. So it goes 1, 2, 3, 4, 5 orders of magnitude, okay? A 100,000-fold increase from before the fillings go in till after they're in. And this is the average weekly mercury concentration in the feces. Now these animals got, respectively, these guys got 16 fillings, and these guys got 12 fillings. So this is the experiment where the fillings are replaced, the red and green lines. So these animals were in-house a very brief time before the fillings went in. Fillings went in, and they gave us this big spike, and then the red and green lines kind of taper down. till out about 60, 70 days when the fillings came back out and were replaced. The process of the replacement caused a spike in the filling, in the GI tract mercury, because you're messing with the mercury and it's going to get swallowed and inhaled. And then they began to go down. It was a very clear demonstration that you take that out, it's the only place it's coming from is the fillings, that it'll go back down. Now, in the later experiment that we did with them, we had the animals in-house a longer period of time. They had 12 fillings put in. The spikes are not as high here for the animals. They had various other procedures done to them, so we've got some other spikes that might have resulted from teeth grinding and stuff. And I mean, not because they were hurt by the procedure, just because they were annoyed by the procedures. They were well treated throughout. But this mercury doesn't go down. I mean, these animals continued to evolve mercury in the neighborhood. This is about 5 to 6 micromolar mercury for the entire time they were in-house. And this is the fecal concentration, okay? Mercury is also going across the buccal epithelium into circulation. It's going entirely through all of our detoxification cycles. And the animals, this is quite striking and not been followed up on, but they've seen this kind of thing before. That these were two cynomolgus monkeys, and these were two rhesus monkeys, and their post-mortem kidney levels for mercury are very, very different. F78, which is the green line, had very little, had 3.3 micromolar mercury in the kidney, and the red line, which is this big spike here, had 98 micromolar, okay? 30-fold higher mercury in kidney in those two animals. And then also this pair, I'm sorry, that's a black line, I guess. This green line had about 9 micromolar mercury at postmortem in the kidney. And that's this green line here, like that. And his R87, separately caged, in fact, these were in the vivarium at different times, had, considerably more mercury, 63.4 micromolar mercury in the kidney at postmortem. So animals can differ very, very much in terms of their loading. And there's maybe genetic aspects of that, there are developmental aspects of that. Monkeys have personalities. I mean, they talk about the ones that just grind their teeth all the time and the ones that like to cooperate and don't like to cooperate, and so forth. So monkeys are a good model. They're a very expensive model and a very limited model in terms of the economics of the situation. But this was basically what we saw coming out of this for our microbiological perspective was the resistance in these animals. Prior to insertion of the fillings, they were just sporadic, less than 20%. But once the fillings went in, then you got these spikes of mercury resistance that came tapering back down again after the fillings were removed. These don't include those latter monkeys where the fillings were not removed. And correspondingly, here are resistance to tetracycline in the purple and ampicillin in the blue. So coming on early on, there's just sporadic resistance. This was from a set of animals that came in from the wild with high antibiotic resistance, but during the time in the vivarium on antibiotic-free food, those were washed out, I mean, by the natural processes of the digestive system. So we had pretty much everybody at 0 when the fillings went in, and the fillings then went in, and we were getting spikes of tetracycline resistance that tapered down, and then it took a while, it kind of delayed a while, but 5 weeks in we got a spike following on of ampicillin resistance, and then that tapered down. So we have looked at many of the bacterial strains. They are transferable. All of this is actually published a long time ago, but the take-home lesson from that was that a non-antibiotic toxic agent can co-select for antibiotic resistance because the loci, in other words, the genetic elements for the relevant traits, are physically linked on mobile elements such as plasmids and transposons. So they can spread throughout the population. As soon as one bug in the population gets one of these things, if the selection is on, they're transferring these things all the time. So it won't take very, very long for that to be transmitted throughout the population. So, to paraphrase the real estate folks, the bottom line is linkage, linkage, linkage. Genetics is very, very important here, okay? Okay, So that was our story. That's how we got into this business of looking at the human side, the health side of mercury resistance. The next thing I'll tell you about is, get that out of the way, the primate intestinal microbes can methylate amalgam-derived mercury. So we had done a lot of sampling of those feces from these various monkeys, and we still had them in the freezer. In fact, some of them feces are still in the freezer. Long after Fritz Lorscheider has retired from the University of Calgary, we still have fecal samples. The next experiment that we went to do was the following one, and this became possible only when the assay of mercury into its different forms became more facile and affordable, because being able to tell methyl mercury from mercuric mercury is hard and expensive, and you'll hear more about that from Chris Shade tomorrow. But this is work that we have done in collaboration with Chris, and part of it's been funded by IAOMT, as was part of the earlier work I described. We got it in our ideas to make one more use of these precious fecal samples that were still in the freezer when Chris Shade developed a remarkably effective procedure for discriminating methyl mercury from inorganic mercury, because we wanted to get very good statistical numbers. That means we wanted to analyze lots of samples. We had 220 samples. And doing that with previously available methods would not have been as sensitive and reliable and very costly. So if you could get anybody to do it. So going into this, we were a little bit suspicious that bacteria in the gut might be able to do this, because the sulfate-reducing bacteria that are the ones that are known to methylate in sediments and lakes and so forth, and they do. They're the ones, the culprits in the methylation in ponds and rivers and lakes. So we know that. Many bacteria can methylate mercury in the laboratory, in landfills and others in sewage settings. Okay, so methylation of mercury is not that weird a thing for a bacterium to do. Previously, several investigators had shown that oral methyl mercury is detectable in humans with amalgams. Okay, and these are people who had not been eating fish, okay, but they had amalgam fillings, and the amount of methyl mercury in their saliva was proportional to the number of fillings they had. And lastly, we knew that sulfate-reducing bacteria are actually common inhabitants of the human oral and intestinal microbiota. So we had a reason to think that if we looked at the feces that we had from these monkeys, that we would be able to see if there was any methyl mercury there and whether or not it correlated with the installation of the fillings. And so this is work that we did with Chris, and we did it to address the question, can amalgam-derived inorganic mercury be methylated by intestinal bacteria in a living animal? Right, so what we did was we did mercury speciation, Chris did this, on fecal specimens from 4 previously characterized monkeys fitted with either 12 or 16 occlusal fillings. In the latter case, they were replaced with a glass ionomer after 2 months. And the total mercury had already been determined by the University of Calgary. And Quicksilver, Chris's company is Quicksilver Scientific Incorporated, redid the total mercury and also did the methyl mercury, which had never been done before. Oops. All right, so I just want to remind you that these 4 animals, this was their mercury spiking profile like this, okay? And these are the figures of what's going on with the fecal, inorganic, and organic mercury in these monkeys. So this is the time course of the experiment, okay? And over here on the left side is the total mercury, and that is in a red line and a blue line. So the red and blue lines are Chris's company's total mercury and the mercury from the Calgary tests, okay? So numbers exactly lie right up on top of each other. So we know that this is a very good analysis, and these were all done double-blind. So my technician arranged them, sent them off, and they came back, and then they were not decoded to me until after the whole thing was over. And so we knew that they were well done technically, and Chris didn't know what to expect. I mean, we tell him a range, but not the exact number. So yellow over here is the methyl mercury. Now it looks like the methyl mercury is as high as the mercuric mercury, but that's not true. Methyl mercury is on this scale over here, okay? And the methyl mercury in this case is in terms of nanograms per gram, whereas the mercuric mercury is micrograms per gram. That's 1,000-fold difference, okay? So a nanogram is 1/1000th of a microgram. So this mercuric mercury is quite high at microgram levels, and the methyl mercury is low, but if we were to plot it on the same scale, it would be down here and you couldn't see it, okay? But you can see that the profile exactly matches the profile of the mercuric mercury. There's one spike here in F78 at that point. So this is F78 and F79, I didn't bring that slide, it looks the same way. F78, the mercury goes up, the methyl mercury goes up, and then they both come down again. Now this is R78, one of the two other animals, and I'll just show you this one. Same thing, we have low mercury by both Calgary and Quicksilver assays. Goes up, goes along and stays up high. And the methyl mercury also is is low initially, and then spikes, and it goes along and it profiles exactly the same way. So these are the correlation curves, and they're actually pretty good given the kinds of samples these are for the increase in methyl mercury as a function of the total mercury. So this is the total mercury in the sample is directly proportional. The methyl mercury is directly proportional. And so why is there mercury here at all before the animals have the fillings? And the answer to that actually is that it's in monkey chow. And monkey chow has about 30% fish meal. And there's enough methyl mercury in fish meal that it's in, and the people who do neurobiology stuff with monkeys are getting kind of concerned about this. So, okay. So the take-home from this is a different view of the source of methyl mercury. Oh, yeah, I mentioned this. Pre-installation methyl mercury is from monkey chow, which contains fish meal. But post-installation, methyl mercury increases 12 to 20-fold, much larger than could be ascribed to any change in diet. And the diets were very carefully controlled. The animals were not suddenly eating 20-fold more monkey chow, okay? So the diet was strictly controlled. All they had for as a source of mercury was fillings. The total mercury increased more steeply than the methyl mercury does, and that's been noticed elsewhere in environmental settings, that mercury can go up like this, and methyl mercury goes up to a certain point, but it never goes up as high as the total mercury, which is a good thing. And we have reason to think that the players that keep the methyl mercury from getting quite so high are in fact the bacteria that have this methyl mercury degrading enzyme that I mentioned earlier that's in the system we study, MERB. And MERB bacteria with MERB have been found in these fecal samples and are routinely found in environmental settings as well. And so it's a reasonable argument that little rascals, bless their hearts, they're doing the best they can to save us if we're going to have mercury around by keeping the methyl mercury much lower than it would be if it were just ordinary biotic processes making it. Okay. Right. The other point I want to make, the actual mercury that's coming out per day, methyl mercury estimated is from the GI tract, the best estimates of what we could make, assuming certain things about how many feces, how much weight of feces and so forth the animals had, would be about a microgram per day for animals with this number of fillings. Well, that's not much methyl mercury. you know, a can of tuna fish will get 40 times that, okay? Now, in a can of tuna fish, that 40 times methylmercury is actually bound to protein. This would be methylmercury that was freshly synthesized, okay? In addition, these estimates that we can make about how much methylmercury is in play here from these archived fecal samples is the low estimate, because it doesn't take into account, even if they were derived from fresh feces, it would not take into account what was absorbed into circulation during passage through the bowel, okay? Our point here primarily is just to make it clear that we have a catalyst at work here. It's a catalytic process that's methylating mercury, and it's in that wonderful fermentative center that's in our body that takes care of us in so many ways, except if we feed it mercury, it will make problems in the form of synthesizing methylmercury. Okay, I'm not going to treat that last point. I have 1 minute left. Can I have about like 4 more, maybe? Okay, I want to do something really quick because I really want to get to this biochemistry. All right, y'all know about the periodic table. Or you did once. This is period 12. This is mercury down here. All these other metals are important, okay? These are what I call transition, the metallic metals here, copper, chromium, manganese, iron, so forth. These are called the alkali and alkaline earth elements or metals. And this is your magnesium and calcium, potassium, and so forth. And these are the non-metals over here, okay? Metals, particularly these guys, the blue and the turquoisey guys, they prefer to bind with sulfur and selenium, the soft, these are called soft metals, and these are called soft nonmetals. That means they share their electrons, okay? It's almost covalent, right? The alkali and alkaline earth elements prefer to bind with what are called the hard nonmetallic elements, oxygen particularly, and these are like ionic interactions. I'm plus, you're minus, we can like stand close, that's fine, but we're not going to share, okay? All right? So they're different modes of interacting. And sulfur is a biggie for mercury, all right? Nitrogen can kind of go both ways, but we won't go into that at the moment. All right. The major target in cells for mercury is the thiol group of the amino acid cysteine and its various other forms in the cell, glutathione, lipoamide, acetyl-CoA, and in proteins, all right? Because so much of biology is based upon metals and thiols, that's why bacteria have to have a MER operon. They're vulnerable, just the way we are vulnerable because we have these same kinds of proteins in our own systems. So one of the things, this is a list of cysteine-containing proteins that are really important. This is kind of like gross anatomy of E. coli. This is an E. coli cell depicted by David Goodsell, who's a wonderful crystallographer and artist at Scripps. And this is just a cut section cartoon. Here is the flagellum, that motor that was making those little bugs in Jess's slides swim around. So that's the flagellum. This is the outer membrane. It's kind of fuzzy. It helps the bacteria stick to things. This is the inner membrane. Think mitochondria, okay? This is the energy generating stuff that every bacterium has. Okay? These purpley things in here are various enzymes and proteins and chaperones that help the proteins fold. And the big purpley things are the ribosomes, the ones that actually make the proteins. Okay? And then the messenger RNA and the nucleoid. Bacteria don't have a nucleus, but they have a compact structure that their DNA is in. The reason I'm making this point is because we're not talking about things just kind of rare stuff kind of sloshing around in a test tube. Water is organized within such a thing, okay? Water's tightly organized in our cells. Nobody's sloshing around, but everybody's kind of cheek by jowl. And there are a lot of proteins. E. coli has almost 5,000 coding sequences for proteins. Only about 2,500 of those are expressed at any one time. We went here to look for sensitive proteins, okay? We wanted to look on a grand scale because focusing down and saying, oh, I think it's this protein or that protein, you know, you could do your whole life doing that, one at a time. You don't have to do things one at a time now. You can do things on what's called an omics scale. Genomics gives us all the genes, And proteomics gives us all the proteins. And what we wanted to do, and are doing in collaboration with folks at Pacific Northwest Lab, the DOE, we're looking at what happens to all the proteins when you whack them with mercury, when you whack the intact cell with mercury. And the tip of the iceberg is, you can do it. I won't go through that. I know you can't see this one, but we look for proteins that have a mercury on them, and we can do that through the method that I just slipped through. But these are selected proteins that we find that have phenyl or ethyl mercury adducts. Now, I should say that we're working with the organomercurials for a chemical reason I won't go into, but we're working with these organomercurials, not methyl mercury, because it's so dangerous. And so phenyl mercury and ethyl mercury, which is in methylate, are proxies for us for methyl mercury. And they're going to be chemically very, very similar. And also, as it turns out, very different from mercuric mercury. And all you can see from here, from there, is a slide with rows of color, okay? So each one of these colors corresponds to a different metabolic function whose proteins we have found with mercuric adducts after treating the cells. We hit glycolysis, we hit the TCA cycle, there's a respiratory protein, carbon metabolism, nucleic acid synthesis, amino acid synthesis, DNA repair, transcriptional controllers, the ribosome is a big hitter, chaperones, and oxygen defense. So proteins in all of those metabolic categories are vulnerable, just like the water transporter that had a cysteine in it that my colleague at an Eastern university, which will not be named, said, oh, no, it wasn't interesting, okay? And part of the problem is it does hit so many things. And what we're sorting out now, and our purpose in going into this, was to sort out the proteins that are most vulnerable. These proteins on this list are highly conserved. Yes, this is E. coli. It's a good working model. But for the categories of proteins that we're looking at here, these are all parts of central metabolism that are evolutionarily very well conserved, including even to their cysteines in us. So this is a way to start with bacteria, find out what the sensitive proteins are, the ones that are most vulnerable to being tagged stably by mercury, and then use those as a tool to go forward to eukaryotic systems, including humans and animals, but we'll go through smaller organisms on the way, as a way to develop biomarkers to look for the proteins that are essentially the canaries in the cage in the mine, that when everything seems to be going right and you haven't noticed that stuff is going wrong, you could look for, that's what a biomarker is for. I mean, that's what it's about, is to look for something and say, has this caught a mercury? Is this protein going down? And we know for all of these what systems they're involved in. And any one of these systems, if it were regularly bombarded with mercury, could take out a substantial function of your metabolism. So we can look actually, the proteins that have structures, we can actually look at where the mercury is in that structure. And we're not looking at the mercury itself, but these are where it would be. We know from our work which of the cysteines in the protein are getting mercurated, and that allows us to see in that particular protein what region of the protein has been endangered. So I think I better quit here. There are other things that we can talk about, but we can come back to that. That was iron-sulfur centers. Mercury whacks a very important component of redox biology. It causes the collapse of the thiol buffering system and also electrolyte imbalance. And I've left these out. We have information on them, but Chris is going to talk more about this tomorrow. And it may take other ligands besides sulfur. And we have hypotheses to test. But thanks for your interest and your support.